direct connection with the food that's passing through. They're going to either add things to the process or process the nutrients themselves. So of course we're talking about the liver and the gallbladder. We're going to talk mainly first about the liver. The liver is found just below the diaphragm in the abdominal cavity. It is primarily on the right side of the body. And it is an organ that's very interesting for a lot of different reasons. And one very interesting fact is that you can actually replace the liver. If you donate part of the liver, it will repair itself and regenerate. Now the liver itself can weigh up to or around 3 pounds in the adult. It's the largest organ that we have. And we're going to talk about just some basic anatomy of it first as we get into this. If you look at this, of course, you want to orient yourself. This will be the left of the person here. So this is, of course, the left lobe. And then the larger would be the right lobe. Most of the liver, of course, is on the right side and down here. Now the falciform ligament is a membrane that will divide the right from the left, and it's fairly easy to identify. And its job will help suspend the liver in place from the diaphragm and the wall of the abdomen. Okay. Now this is a view of the liver from behind. You don't have to imagine that you're standing behind the person. So if you get oriented as far as directions here, this is actually the right this time, and this is the left. You are behind the patient looking at their liver, and it's slightly tilted. The liver is more of a wedge shape, so it's kind of an unusual shape. So you want to spend a little time with the actual models in the lab to get this kind of perspective. So again, if we're orienting ourselves, this of course will be the right lobe, the left lobe. Okay. Some other easy things to identify will be the IVC, the inferior vena cava. This is the large vessel that's bringing venous blood from the lower half of the body, and it will take it to the right atrium of the heart, which will sit very, very close to the liver. This is also another thing that's easy to identify, the gallbladder. Now we have two other quadrants that are, not quadrants, but lobes. Two lobes of the liver. They're a little bit harder to identify. This is the quadrate lobe, and this is the caudate lobe. Okay. The easiest way to tell these apart is that the quadrate lobe looks kind of like a quad, like a four-sided structure. So the quadrate lobe will be next to the gallbladder. The other structure that you want to be able to identify on the posterior side is the round ligament, or the ligamentum teres. And this is not the same thing as the falciform ligament, which is in the front or the anterior view. This is a separate structure, and it's actually the remains of your umbilical vein. So in fetal life, when the blood from the mother is coming in through the umbilical cord, the umbilical vein will take the mother's venous blood directly to the liver. Part of it will be going to the fetal heart, part of it will be going to the fetal liver. And this, after birth, of course, will shut down and collapse, and we'll see it as the round ligament. Okay. Now, another unusual fact about the liver is that it is the only organ that actually receives venous blood. That's kind of a backwards kind of thought. If you remember, hopefully you learned a little bit about this when we talked in the circulatory system, but venous blood is actually entering the liver, and it's coming from 3 different sources. And if you think of it as cis, these are the first letters of each of the veins that join to form the portal vein. The splenic vein is bringing venous blood from the spleen. Okay. The inferior and superior mesenteric veins are bringing venous blood from the intestines. They are bringing in all the nutrients that you just absorbed from your food, and they're taking it to the liver for processing. So everything will be shunted into this one main blood vessel. Sometimes it's referred to as the hepatic portal vein, the main portal vein. You'll see sometimes different ways of referring to it. Again, anytime you see hepatic, it's a good idea to already know that that is referring to the liver. You'll see a lot of words that have that prefix in it. One may be hepatitis. You may have heard of that, inflammation of the liver. So again, we have a very unusual circulatory system where we're bringing venous blood into the liver. Now the porta hepatis, this is the name for the entry-exit point of the liver. It is sometimes referred to as the gateway because this is where things are coming in and going out. Now I like this slide because it color coordinates things. We've got the colors that will hopefully help you organize what is going in and what is coming out. First, if you want to start with the hepatic artery, this is an easy one to think of. It's showing you that it's in red because it's carrying oxygen. So this is normal oxygenated blood being carried by an artery that goes to an organ. This is how it works in every part of your body. And of course, here's the difference. The portal vein is bringing venous deoxygenated blood into the liver. So again, these arrows are really good. They're giving you direction of flow of something into or out of the liver. Now, in green here we have something called bile. We'll be talking in more detail about what bile is. But bile is actually a greenish substance that's produced by the liver that helps us to break down fat. So it'll actually be leaving the liver at this point. So this is, of course, an entry, exit. All right, we have things going in, we have things coming out. So these three vessels will be called altogether the porta hepatis. Now, of course, blood has to exit the liver, and it's going to do that here. This will be a typical venous system. Smaller veins will merge to form larger veins. Eventually, you get the right hepatic vein, the left hepatic vein, and they will merge into the IVC, which, of course, we know drains the venous blood from the lower half of the body into the heart. Okay. All right, now if you look at microscopic anatomy, we're looking down at the level of the cells themselves in the liver. The liver is fairly homogeneous. If you were to cut through the liver at any part, you're going to see this fairly repetitious liver lobules that all basically look the same. They have 6 sides with a central vein in the center. So this is what we call the functional unit of the liver. This is where the actual work is being done. And all of these little dots here are representing the hepatocytes, the liver cells that are going to do the actual work of the liver. At each corner of this liver lobule is something called a portal triad. Triad, of course, is referring to the fact there are 3 vessels at each corner, and they're going to be made up of, every time, a small branch of a bile duct, okay, because you've got to have bile being produced. You're going to have a very small hepatic arteriole. Okay? You're going to have a branch of the hepatic artery, and that's, of course, bringing fresh, just regular blood into the organ. And then you'll have a branch of the portal vein that, of course, is bringing in venous blood, carrying nutrients and things that need to be processed in the liver. Okay. Now, what does the liver do? Well, it does a lot of things. I'm just going to touch on a few of the major functions of the liver. When you first start thinking of the liver, you want to think this overall picture, that it processes things. I like this picture because it shows you that it's like a factory. We're taking things in, we're changing them, we're rearranging them, we're making things and sending it out back into the body. So it's going to be processing all these nutrients that you took in your food down in the small intestine and carries it to the liver through the portal vein, and the liver will process it. It also plays a role in getting rid of things we don't want. Toxins, chemicals, drugs, things in the blood that we want to get rid of. So it's going to be, overall, you want to think of the liver as this processing metabolic site where we create things, make things. One function, any fat-soluble vitamins that we have taken in our diet, we can store a certain amount in the liver for later use. We're going to take amino acids that have been absorbed and make proteins. In specific, we can make the plasma proteins that are involved in blood clotting. Those are made in the liver. We also form albumin here. This is the largest protein in the blood, in the plasma, makes up the majority of plasma proteins, and it's involved in helping maintain the osmotic pressure of blood. The liver also, at any given time, is going to contain at least 10-15% of your total blood volume. This means it acts kind of as a reservoir for the blood. In case you have a severe loss of blood, the liver will be stimulated to release some of that and help. And the reverse, of course, is true. If there's too high a blood volume, the liver can absorb some of those fluids. All right, we're looking back at a view of the liver lobule again. We're looking at here the portal vein is bringing in venous blood, and as it filters in through this liver lobule, it comes in it gets small enough that it gets down to this level at the edges of the lobule. And this will be the portal triad, and it will again branch into really, really small sinusoids, which are basically leaky vessels. They're going to allow the blood to flow over these hepatocytes. So the hepatocytes are arranged in these this nice pattern of cells, and they're going to be doing all the actual work, all the processing of nutrients, removing toxins. And as the blood filters through here, we're also going to have an immune protective role. Sometimes there will be bacteria in the blood, other types of antigens, and macrophages will be sitting here in these little blood vessels, and they will, of course, process and digest anything foreign. These are also known as Kupffer cells. Okay. Now, another important function of the liver is to help us regulate our glucose levels. You may remember back in the endocrine system, we talked about insulin, about how insulin tells the cells to take in glucose that is in the blood. That's a huge function of insulin. But the liver is also playing a huge role in this regulation as well. If we have high levels of glucose in the blood, we've eaten a huge meal, a lot of candy bars, whatever we've eaten. Insulin, of course, is telling the cells to take up the glucose. Insulin is also telling the liver to take it in. So insulin is also going to signal the liver to take this glucose and store it. And we store it as something called glycogen. If you look at this picture, you can see this is an individual glucose unit. So in the liver, we take these glucose units that are in the blood, we bind them together and form something called glycogen, which is this long branch chain of glucose units. And we can store it for later use. Okay. Obviously, we're going to store it in the liver. We've got ready access to it. And the other place is going to be the skeletal muscles. And if you remember back from 201, you use this as an intermediate energy source if you go and do some kind of exercise. So anytime you see this word, glycogenesis, genesis, of course, means creation of something. And in this case, it's creation of glycogen. Okay. Now, we've stored glycogen in the liver. Let's say for some reason all of a sudden you don't have enough energy. You're starving, perhaps. You need a little bit of extra glucose. So the body is going to signal the liver to break this glycogen down. All right, our signal, of course, is another hormone, glucagon. This is produced in the pancreas. You might remember this from the endocrine system. It will tell the liver to break down glycogen into, of course, its subunit, which is glucose. It gets released into the blood, and your blood glucose levels increase, which is what we want. We want to get the blood glucose levels back up to normal, which is homeostasis. We want to maintain a normal balance. So glucagon, of course, will be causing your glucose levels to go up, and this is in opposition to insulin. Okay. All right, another reminder here, anytime you see lysis, that's a word that means to split something or to break it. So if you saw glycogenolysis, which is this process, it means we're breaking down glycogen. So it's always a good idea to understand some of the suffixes and prefixes as you learn anatomy. Here we go again, gluconeogenesis. Genesis meaning creation. So sometimes the liver has to actually make glucose. If you are starving, if your body is not getting any energy in, it's going to get glucose. Glucose is the only carbohydrate that can get into the cells. So the cells have to have it to live. So we're going to make glucose somehow. So the liver is going to be the area that we do this. So an easy way to think of this is fat. So let's say you have stored extra glucose as fat. We've stored this, we have this nice supply. You can break this down into fatty acids. The liver will take those fatty acids and rearrange it and produce glucose. And again, secrete it into the bloodstream and help raise your glucose levels. Again, glucagon is also stimulating this process. Now, if you are starving to the point where you have used up all your fat reserves and there is nothing left, we will turn to the muscles. Your muscles will start breaking down the proteins that are structural proteins. Amino acids can also be converted by the liver to glucose. We just simply rearrange the elements, the carbons, the hydrogens, and we create glucose, which is what we ultimately need for the cells. Okay. Another thing the liver is doing is getting rid of things we don't want. As we're breaking down proteins in the diet, as we're maintaining protein in our muscles, we are going to be breaking down amino acids. And one of the parts of every amino acid is nitrogen. And it's very difficult for the body to get rid of nitrogen. In its simple form, the nitrogen molecule will bind with hydrogen and form ammonia, which of course is very toxic to the body. So very quickly, the liver is going to take two ammonia molecules, combine it with oxygen, and create urea. And then the urea that is then carried in the blood will be filtered out in the kidneys, and it will be excreted in the urine. Okay? So, of course, this ultimately started in the liver. Okay? Just like ammonia, we also have other things that are being carried by the blood that we don't want. We can think of them as toxins, drugs, but they may also be drugs that you're taking, an antibiotic, something for pain, some kind of medicine that the doctor gave you. Anything that is in the blood, the liver is going to have a role in breaking this down. That is just a normal process. Otherwise, well, basically the liver is going to maintain the balance of things, and it's getting rid of any abnormal chemicals in the blood. One, of course, is ethanol. Person drinks lots and lots of ethanol, it will get converted in the liver to more innocent byproducts that we can use in other ways. And of course, over years of abuse, liver cirrhosis, this is scarring of the liver that can occur due to too much drinking of alcohol. The liver simply can't cope with the amount of alcohol it's having to process, so the tissue becomes scarred over time, and the liver stops functioning. Now, the major digestive function of the liver is to produce bile. Okay, all these other functions are again related to the digestive system, but the main one is here, bile production. And I've already said that it has something to do with fat in the diet. You're producing almost a liter a day, every day, of this particular substance. So what does bile do? When we take in lipids or fats, they are hydrophobic. They do not like water. And our human bodies are made up about 60% water. So we have a difficult time absorbing fat because it doesn't want to mix with us, which we're, of course, water. So we have to have a way to get around this, and that's bile. Bile will emulsify the fats. And the word emulsify means that I break this fat down into smaller and smaller particles. It's not digestion. Digestion implies a chemical breaking of bonds, and bile does not do that. So you want to be sure that you understand it is not an enzyme. This is not digestion. Remember, chemical digestion means an enzyme is breaking a chemical bond. Bile does not do this. It simply coats the fat. And if you look here, you'll see all these ingredients. Bile is composed of several different things. Bile salts, triglycerides, cholesterol, pigments. It's a mixture of things. And when it happens to come in contact with the fats, the hydrophobic tails of these little phospholipids will orient themselves toward the fat. And of course, these little head parts will have a charge, and they will orient themselves toward the water. And so what you're doing is creating smaller and smaller particles that are surrounded by water that will get us to the point where we can digest the fat. So the next step has to be enzymatic digestion or chemical digestion. So bile is going to emulsify it. We're going to make these little small particles that are coated with bile. They're still not small enough, though. In the small intestine, you're going to also have enzymes called lipases. And that word, of course, is coming from lipid. Right? That's where the base is. And so anytime you see ASE, you're thinking of an enzyme. So this, of course, would be an enzyme that breaks down fats. The pancreas makes these. At this point, thanks to bile, it's small enough that these lipase enzymes can act on it and break the fats down into their smaller subunits, the fatty acids and the phosphate parts. So these fatty acids are finally small enough that the cells here in the small intestine can absorb it. Things have to be small enough to literally go through these cells. It's still too large. The fatty acids still cannot be absorbed by the bloodstream. The capillary that's here in the the villus is still cannot absorb it. So we have to have a special system to absorb fats, and that's actually a part of the lymphatic system. Okay, so if you remember way back from the circulatory system, we mentioned the lymphatic system. So in the villus, in the small intestine, you learn a little bit more about that when you get into the small intestine, but within this villus is a lacteal. It's going to contain a capillary network to absorb other kinds of nutrients, but the fatty acids are going to have to go into this special structure called a lacteal. It'll pick up the fats, carry it through the lymphatic system. Lymphatic system eventually drains into the venous system, and you're back into circulation. Blood will then take the fats through its network and carry it to the liver, and then the liver will process the fat, break it down. If the body doesn't need an immediate source of energy, it will store it as adipose in the tissues. All right. The other accessory organ is the gallbladder. The gallbladder's job is to store excess bile, to concentrate it, and then to release it when you need it. So if you think about the liver, again, liver is producing the bile in these tiny little liver lobules. So each time it flows into the bile duct, those bile ducts join until they form larger and larger ducts, and they form a network that's taking this out of the liver. So eventually all the bile ducts will drain into the right hepatic duct and the left hepatic duct. Okay? Okay. Those two join to form the common hepatic duct. Anytime you see the word common, it's telling you that two vessels have joined. And another key here will be to understand that if it's a duct, it's not in the circulatory system, correct? It's not a vessel of that type. It is carrying some type of product somewhere in a tube or a duct. So it has nothing to do with being a vein or an artery. Okay? So at this point, the right and left hepatic ducts have joined. They're carrying the bile out of the liver. They have, at this entry point, at the porta hepatis, it is now technically called the common hepatic duct. All right, so let's say we've produced a lot of bile. We haven't eaten a lot of fats in the diet that particular day. We don't need the bile. So all the excess is going to be pushed over here into the gallbladder through the cystic duct here. And we'll store it in the gallbladder until we need it. Now, if we need the bile, it's going to flow through the common bile duct. After the cystic duct, the name changes. It becomes the common bile duct. And at this point, it will carry the bile down to the small intestine, and it will go out of an opening called the hepatopancreatic ampulla. You may remember this from studying the pancreas in the endocrine system. So this is going to be the opening that allows the bile to enter the small intestine. Now, how do we know when to let the bile out? Is there a signal? And, of course, it has to be some type of hormone. So cholecystokinin, or CCK, is the hormone that's going to help us regulate the release of bile. And, of course, that's going to be triggered by fat in the food. You've eaten a large meal, say it's nice pizza with lots of fat. The stomach mixes it. It then allows the food to move into the small intestine. The first part is the duodenum. So when the food enters this duodenum, the body is going to sense the presence of high amounts of fat, and that will trigger cells in the duodenum to release CCK. So CCK, of course, is a hormone. It will travel by the bloodstream. It will do three major things. First, of course, it's going to tell the gallbladder to contract, to release this bile, because we need it. It's going to also tell the pancreas over here that we need some digestive enzymes. We need lipase for the fat, right? Okay. We also, obviously, we've got food, so we're going to need all the other enzymes as well. But pancreas will be told that we need a little bit of this released, and then we've got to get it out. We've got to tell these sphincter muscles to relax. Ordinarily, unless there's a signal, the bile will stay backed up. It will not come out into the small intestine unless there is a hormone signal. So the bile will be backed up all through the common bile duct and, of course, stored in the gallbladder. Now, sometimes things go wrong. Gallstones is a very common problem with people. The whole other name is cholelithiasis. Chola is referring to gall, and lith means stone. So sometimes we get these crystallized stones within the gallbladder. Of course, they can be extremely painful. Anytime that the gallbladder tries to contract and release bile, it squeezes on the stones, creating this agonizing type pain. It can be due to the fact that you're just simply not making enough of the bile salts. You could have too much cholesterol in your diet. This is someone who's overweight, has a high-fat diet, higher association with diabetics, some type there. And it may be that the gallbladder is just not functioning correctly. It's not able to squeeze out the bile as it should. And in the end, everything crystallizes and get these stones. Usually treatment, sometimes they'll surgical removal if it's maybe just one. Sometimes they will use sound waves to try to break them apart. Typically they remove it, and this is cholecystectomy. Anytime you see the word ectomy at the end of a word, that means I have surgically removed something. And again, chol is referring to gall, and cyst is for the bladder. So cholecystectomy is a very common surgery these days. Can you live without your gallbladder? Yes. This is very common. What they will do is they will go in and simply remove the gallbladder here, and at the level where the cystic duct joins. So what they have left in place, this is the common hepatic duct. at this point here, it becomes the common bile duct. And over time, this particular duct system will just simply enlarge. It will expand, and it will basically act like a gallbladder. It will hold this extra bile, and when you need it, it will release it. So it will function as a gallbladder in a sense. So yes, you can live without your gallbladder. All right, that's all I wanted to talk about today. So I hope you've enjoyed learning a little bit about the liver and the gallbladder, and thank you very much.